My name is Luiz Rocha. I'm the curator of fishes at the California Academy of Sciences. Collections are in the news in the last few days because of what happened to the major national museum in Brazil. The loss is almost incalculable for many reasons. The museum is it's exactly actually 200 years old. The National Museum in Rio was the first museum I've ever visited. I've always had this passion for fish and science, and if you have this passion, the museum is the best way to visit to kind of feed it. So collections, they kind of almost work as libraries. So every one of these jars that you guys are seeing is almost like a book. It contains a lot of information, not only on the labels themselves. So in the label, we kind of put all of the information of where the specimen was collected, by whom, when, and in the associated electronic database, we have other information about the specimen, but also we have the specimen itself that can be used over and over again for research for multiple different reasons. If someone wants to figure out what this fish was eating at that time, they can come and look at their stomach contents. If they want to look at their morphology and see how it varies between location, they can come and look at it. So because it's such a treasure trove of information, collections worldwide, they exchange specimens constantly in an aim to become more diverse and more useful for the scientific community. So in the 1940s, what happened was that we received a lot of specimens from the museum in Brazil, from the National Museum from Brazil, the exact one that went up in flames. We received a lot of specimens from them to make our collection a little more diverse and more useful for science. This is one of them. This is a grunt, is a group that I worked with. This one was collected in Recife, which is a town that is just south of my hometown in Brazil, just about 60 miles south of my hometown in Brazil, in northeast Brazil. It was collected in January of 1945. So even during years of war, people don't really stop their science. They keep collecting and they keep doing things. And it made its way from the museum in Brazil to here. And then here it gets studied and understood, and it's used for science throughout the world. So this one is kind of a sad story that also highlights the uniqueness of biological collections. I've never seen it before, but the story just strikes me in a way that it gives meaning of everything I'm doing. So this is Azurina upalama. It's a fish that was collected in 1903 in the Galapagos. It was described from then. Back then, we have field notes saying that it was a relatively common and abundant species. It's a homocentrid, a damselfish that feeds on plankton, so it lives high in the water column and feeds on the tiny microorganisms that live in the ocean. And it depends highly on nutrients that are in the water, because when there are no nutrients, there's no plankton, which is its food, and then when there's no food, they die. So this fish was collected in 1903. It came here to the collection. It's a holotype, which means it was the specimen that was used to describe the species. Now, as I said, it's a species that feeds on plankton and lives in somewhat open water in the Galapagos. And it was off and on seen throughout the 100 years almost after it was described. But then in 1982 and 83, there was a very strong El Nino. So El Nino is that warming of the ocean waters. And with the warming, it comes the lack of nutrients. So warm water usually doesn't have any nutrients in it. It's a really clear water. So that pool of warm water brought by El Nino, it sat on top of the Galapagos for almost 3 years between 1982 and 83. It was a typically warm El Nino. And then after that El Nino, this species has not been seen in the Galapagos anymore. And it was it was a relatively common species in the Galapagos. So it's what we have right now, the closest that we have right now to being a marine species that went extinct. And all of the known species of this, or all of the known specimens of this species are right here in our shelves.
Wow.
So this shows you how unique a collection is, and that this is the only place basically where this species lives right now.
And does that extinction have any kind of implications for things that you're studying today as conditions change in the ocean?
Yes. So the same thing that kind of drove this species to go extinct is what's driving a lot of species to be threatened today. It's basically climate change. In that particular case, it was El Nino, but we know that climate change is causing El Niños to become longer and more intense, and it's now highly damaging. It's the number one threat right now to coral reefs, and coral reefs happen to be the ecosystem that I study. So I'm seeing through my lifetime. I've been going to the field for the past 30 years or so. I've seen with my own eyes basically the decline of coral reefs worldwide. I study fish, but when I see a coral dying, I know that's fish habitat that's going away. And with it goes the fish, it goes a number of other organisms that associate with that ecosystem, and it's declining at a really alarming rate.
So these kinds of records are even more critical as we kind of try to understand and prepare for what's coming.
Absolutely, yes. We need to know where those species were in the past. We need to know how they interacted with their ecosystem, if we're even to try to restore those habitats at some point.